Welcome to Brain Ponderings. I'm the host, Mark Matson. Today, my guest, it's a real pleasure to have Ziv Williams with me. He's a neurologist and neurosurgeon who's doing some remarkable work recording from individual neurons and collections of neurons in the brains of awake humans and non-human primates. And he's doing some really fantastic work that's getting to a problem that has been previously very hard to tackle, particularly in humans and non-human primates, and that is social cognition, and which occurs in a lot of animals, but then language, which is, as far as we know, at least in the detail, we use language unique to humans. So welcome, Ziv.
Mark, thanks so much for having me. It's a pleasure to be here.
And what's your early background? I see you did got an undergraduate BS in biochemistry or chemistry and cell biology at University of California, San Diego, and then you went to med school at Stanford. Where did you grow up, and were either of your parents physicians or scientists?
Yeah, I grew up in California primarily, as a kid, and then later on stayed there before moving to Boston. My parents were a mix. So my mom was a scientist. She was very interested in molecular biology, and my father a freelance writer.
Oh, interesting. Yeah. Did he teach you any writing skills?
Not really directly.
Okay, so you developed those more on your own. Okay, so you're a neurosurgeon, and you're currently at Harvard Medical School at Mass General Hospital, and you have appointments, well, in several centers and initiatives there. Also, you interact with people at MIT, which is close there in Boston. Can you talk a little bit about, okay, so you went to medical school at Stanford and you're a neurologist, and most neurologists, like, they focus on a specific disease or an injury to the nervous system. How did you get interested in looking at normal brain function in humans?
Yeah, it's one of those things that just kind of comes out by accident. When I was rotating in medical school, I became very interested in neurology and then in neurosurgery. Part of it was because there's still very little that is understood about the human brain at a very basic level. And there's also an opportunity within neurosurgery to treat patients with really disabling disorders. And so just rotating as a medical student, it really struck me how much we can potentially do, but also how much is still not really well understood. And it's kind of an interesting awakening when you're developing as a medical student. You have this assumption that everything is already known and you just need to, somebody needs to teach it to you. But there's still so much that's really not well understood, and there's so many disorders that are still fairly disabling, and we don't really have good treatments for them, like depression, Parkinson's disease, paralysis, many forms of degenerative neurological disorders. So it's a really exciting field.
Uh-huh. And when you went to Boston, how did you learn the electrophysiology? I mean, these are not trivial. So you've got to implant electrodes in the brains of humans, and of course, there are a lot of interventions now, like Parkinson's. You mentioned deep brain stimulation. There are some treatments based on stimulation, but that's maybe, well, I don't know, maybe it's easier in some sense than actually recording from neurons. And can you talk about, how you developed those methods? Who did you work with that taught you how to do that?
Yeah, I was lucky enough to have really phenomenal mentors, both here in Mass General Hospital and at the Harvard Medical School. And so my mentor here at the time was Ahmad Eskandar, and he was a functional neurosurgeon. Was very interested in also some of these really basic questions. And he trained as a basic primate neurophysiologist. So he learned most of his electrophysiology skills in rhesus macaques. And he was able to translate a lot of these basic techniques into human electrophysiological in the human electrophysiology settings. And then also was lucky enough to have a really phenomenal mentor at the medical school when I did my postdoctoral training, John Assad. And he's just an absolutely phenomenal basic neurophysiologist who really knew how to ask really difficult questions and do it in a really smart way. And he really taught me how to think about neurophysiology. What are the questions that are important to ask? How do you control for all the variables that you can encounter when you're asking these questions? And how to be very just rigorous and robust about the analysis that you're doing. So I was lucky enough to have people to really teach me these really fundamental skills.
And, looking back on your publications, so you've been doing this a long time, more than 20 years. And was your first studies in the rhesus macaques, the monkeys, or humans, as far as actually implanting electrodes and recording?
Yeah. My earliest experience had really been in primates, learning some of the core skills of how to do really good electro neurophysiology, and then rapidly translated into intraoperative human neurophysiology. So I worked very closely with Dr. Eskandar as a resident when I was training to learn how to do neurosurgery. And we had developed techniques together for translating some of these basic skills into the intraoperative settings to try to study questions you can really mostly study in people, but not very easily in animal models.
And that's where the social decision making and the language come in as two really largely unknown what's going on at the levels of individual neurons and neurals, local neural circuits, or maybe even, broader communication between brain regions. So from a technical standpoint, so you're using, so in my lab, we did a lot of work on cultured neurons or brain slices, and we'd put one electrode and one neuron using whatever patch clamp or other techniques. But my understanding is that you're implanting multi-electrode arrays in the brain so that you're essentially recording the firing, the action potentials of individual neurons, but you're recording from an array. Is that right?
Right. So the basic idea is similar. So we're using these really miniature antennas to try to pick up these signals that are being produced by individual neurons. And more recently, we've been able to adapt these multi-electrode arrays that have many hundreds of these little antennas that are able to listen to the chatter of neurons as they're talking to each other and doing what they're meant to do. And by being able to record from all these neurons simultaneously, we're able to build a much more detailed picture of what all these individual cells are doing as we're doing these really basic cognitive tasks like language or social cognitive behavioral tasks and things like that. So it's a very unique opportunity, and it's opened up the door to a lot of really interesting questions.
Now you mentioned you're listening to the neurons. Do you actually have speaker hooked up so you can hear the neurons firing?
Yeah, so we can actually both listen to them, and we can actually see their activities in real time as they're happening. trying to understand what is happening in real time is much more difficult. So a lot of the analyses we do after the fact, but we can actually hear them and look at them in real time as we're recording.
That actually helps a lot, right? Because you're doing neurosurgery, you're moving an electrode, in the brain tissue. And so I would assume you're focused on that with your eyes and your hands and stuff, but then you can be listening to what's happening as you move it. It probably helps quite a bit.
Yeah, and these electrodes are incredibly fine. They're they're about the size of a human hair, and they only go into a very superficial part of the brain, very for a short period of time, and then we simply take it out. And this is in an area in which we're already planning on doing surgery for clinical purposes.
Yeah.
So it's done in a way that doesn't really, perturb significantly what we're planning on doing for clinical purposes,
Yeah.
but it lets us, for a short time period, allows us to listen to what the brain cells are doing, and allows us to ask very interesting questions about it.
Yeah. And yeah, so the very fine electrodes, of course, I think most people listening may know this, that there's no pain receptors in the brain.
Correct.
So the person is not experiencing any pain. They're not having meaningful tissue damage. And a lot of this is in epilepsy patients, right? Where you use electrophysiology to determine the brain region where the neurons are just firing out of control, because that's the tissue that needs to be removed, so that then the person, hopefully, if the surgery is successful, will no longer have seizures, or that will be reduced.
Yeah, no, that's exactly correct. So we'll do it in situations like that. And also in cases in which we're putting electrodes already for other purposes, like for treating Parkinson's disease or essential tremor. So it's already integrated in as part of the clinically planned electrophysiological recordings. And it, like you said, it does give a unique opportunity to record briefly within that setting.
All right, so can you talk first, as far as experiments, about your work on social cognition, social decision making, what brain regions or region you're looking at, why, and then maybe the best way might be if you can just, I'll kind of I'll refer you to like some publication you've had in the last few years, multiple Nature papers, a science paper, but and then maybe kind of go through those, because you're not just recording, you're doing, for example, in the monkeys, you're doing some manipulation of their social interactions. So maybe start out with the social cognition aspect and how you went about developing kind of a paradigm where you can actually control social interactions while you're recording.
Right. Yeah. No, so social behavior it's a very interesting thing. We all do it. It's natural, and it's really fundamental to our ability to interact effectively with others. And primates and humans are especially very social animals. We do this all the time. We're doing it currently. We're interacting with each other, and it's very important to be able to try to understand what somebody is thinking or feeling. And it's a complicated thing to do when you think about it, because at no point do you have access directly to my brain. You don't truly know what I'm thinking. But it's something that you're constantly trying to do. You're trying to ask yourself, well, does this person like me? How can I interact in a better way to make this interaction smoother or more, beneficial potentially? This is something that happens also routinely in economic arenas, trying to understand economic markets. Economic markets a lot of times are driven by interactions through many agents. This happens in political arenas, but I would say most importantly in day-to-day interactions that we do all the time. But trying to understand how this works is not trivial. It's a little bit different than how we've traditionally studied a lot of cognitive processes. So things that are a little bit simpler, for example, if I press a lever, I'll get reward, or if I see a dot on the screen, I need to move my arm in a particular direction. These are called sensory motor behaviors, where there's a sensory input and you have a certain type of behavior. In social behaviors are much more dynamical. My behavior may affect somebody else, but my effect on somebody else may affect how they think about me, how they interact with me in the future. But even beyond that, a lot of us interact within these really complex groups where I may, for example, offer or give a favor to one individual, but that individual may reciprocate that favor to me. And then you have a third individual that's left out, and they may get unhappy about the whole situation. And so it's a really interesting construct. And so one of the very earliest things that we wanted to try to ask, and this isn't a primate model, rhesus macaques are very social animals. They're primates, they live in groups, they constantly interact with each other, and they have these really interesting hierarchies. And so we wanted to start asking a really basic question about, are there particular neurons and parts of the brain that are thought to be involved in social behavior that are uniquely tuned to encode information about somebody else's thoughts or actions? And so we focused on a part of the brain. It's called the dorsomedial prefrontal cortex, or the anterior cingulate cortex. It's right in front, the front part of the brain, and right in the center, close to the center. And it's been shown to be involved in things like autism spectrum disorder and depression. It's very involved in social behavior. And so we recorded the activity of individual neurons in two animals, two primates, as they interact with each other. They're playing this cooperative game called the prisoner's dilemma game. And it's a really interesting game. You may have heard about it. This is something that was talked about, for example, in the movie A Beautiful Mind.
Yeah.
To try to understand how individuals make decisions when it comes to interacting agents. So we actually had these primates play this game with each other. And just for the audience, the fundamental premise of this is essentially that two individuals can cooperate with each other, and that would be the most mutually beneficial interaction. But if one person cooperates and the other defects or decides to cheat, for example, the one that defects and is the cheater actually gets a much higher reward than the one that tries to cooperate. But the linchpin here is that if both defect or both are trying to cheat each other, then they'll get a much lower reward than if they both cooperated. So it's a simple matrix, like a 2x2 matrix, but it brings about these really interesting potential interactions where you can cooperate with each other, you can cheat. And the critical part of this game is you really need to try to have a representation in your head of what somebody else is likely thinking, even though it may not be apparent. And we found that there are neurons in this part of the frontal part of the brain that fairly reliably track and try to predict what somebody else is thinking, even though that those beliefs or thoughts are really hidden. They're not apparent. There's nowhere where the animal is explicitly saying, "I'm going to cheat right now, and you better beware." So it's really interesting to find these, and they kind of complement these canonical mirror neurons that have been known for a long time, where there are certain neurons in parts of the brain that will mirror exactly what you see somebody else doing, but they don't really differentiate between self and other, and it hasn't really been clear whether they can encode information about somebody's hidden beliefs that are independent of their actions. So this was one of the very first things that we did that were very interesting.
Okay, so so these neurons in the prefrontal cortex, then in this paradigm where you have the three monkeys essentially sitting around a table, and you're recording, from my understanding, is one. So you've got electrodes in one of the three, but
Right.
then you vary as you described, you vary the reward or not thing. But so you find that you can identify neurons that they're active before that monkey makes a decision as to whether or not it's going to give one or the other monkeys a reward. Is that right?
Yeah, that part was a little bit of a kind of a related study. The first one
Oh,
was more two pairs of animals that just interacted with each other. This one is even more interesting, I think. It was where we had three animals all
right.
simultaneously interacting with each other. And that brought about like really interesting questions, because it kind of forces you also to not only have a representation of any other individual, but somebody else specifically. So am I interacting with Bob or with
Yeah.
Sam? Or, and it's important, right? It's important to be able to have a map in your head of, who exactly is who. Um, and the interesting finding with that particular study was that we found neurons in a similar part of the brain, in the prefrontal cortex, that not only tracked what other individuals were doing, but who specifically did what to whom and how. So it kind of, you were able to glue all these neurons together and build a really detailed map of who was giving reward to who was reciprocating. Um, and you could, it was, I would say, kind of a very simple uh, social network map. You can kind of think about it as, type of Facebook in your brain, or a kind of some other, social app in your brain that just keeps track of everybody and what they're doing. And it's important when you think about it. If somebody, for example, keeps on uh, misbehaving towards you or even others, these neurons even track what others and other individuals were doing to each other. It is important for you to be able to have a representation of that. So it's kind of interesting to see these cells in the brain.
Now, is it possible with the electrodes you're using to stimulate and see if you can affect the interaction between the animal?
Yeah, those are other really interesting thing. We could actually modify their behavior very specifically. And so we could actually make the animals less likely to cooperate, even though we weren't changing any other aspect or any other fundamental aspect of their behavior. So by disrupting this very small node in your brain, we were able to make the animals cooperate more or less. And we could disrupt specific aspects of social behavior. Um, and in another unrelated study, this is more in mice, and we can talk about it later, we could actually make the animals more social. So animals with an autism spectrum disorder type phenotype, we were able to actually uh, turn them from fairly asocial individuals to fairly social individuals. So it's an interesting part of the brain that's very centrally involved in many types of social behaviors, and we can modulate it to make individuals less cooperative or more cooperative, or less social and more social. So it's really interesting that you can
Yeah.
do that. Yeah.
Yeah. Well, go ahead and talk, because autism spectrum disorders are, it's there's a lot of kids affected, right? And one of the diagnostic effects is that person kind of doesn't like to interact with other people, and they tend to do repetitive behaviors.
Right.
And so go ahead and talk about the mouse studies. So there's certain mice that that are genetically altered in ways that cause them to behave like they have autism, or at least in terms of social withdrawal, right? Is
Yeah.
that
So yes, autism spectrum disorder, it's really a spectrum. And there's certain genes that have been associated with it. So one of them is called SHANK3, and it's been found in about 1% of individuals who meet the criteria of autism spectrum disorder. And the interesting thing is, and this is something that you can do in a mouse model, we've developed a lot of very advanced genetic techniques that allow us to do this. And you can actually knock out that gene in a mouse. And the interesting thing is that they will have a very specific social behavioral deficit. They'll become very poorly social, and mice are actually very social animals. They like to be with other animals of their same type, and they'll lose that preference. But the interesting thing that you can do, and this is using a variety of genetic techniques, you can actually knock that gene out, but you can also restore it. And you can restore it in an adult. And the interesting question that we're trying to answer was, can you have an adult that has their whole life had an autism spectrum disorder type phenotype? Can you actually reverse that? And so we did a number of different things. The first was where we restored gene expression throughout the whole brain. And we found that we could actually make the animals not only more social, but even more social than their original, what we call wild type counterparts. So animals that grew up absolutely normally. And the other really somewhat surprising finding was that we could also restore that gene very focally in part of the medial prefrontal cortex, so the same area that we've been talking about. And even when you just restore genes locally to that area, you could actually make the animals more social as well. So you're able to recover certain aspects of autism spectrum disorder. Now, we didn't touch on other aspects of this disorder, like you said, repetitive behavior, and sometimes individuals are a big component of this disorder is also sometimes language disability, and this is something that we can't really test in mice. But it did give some hope. As a neurosurgeon, we may have the opportunity to focally restore gene expression in one local part of the brain, and that could potentially lead to avenues of recovering certain behavioral deficits like social disability. But it's still a very complex and very heterogeneous disorder. So this is just a very small initial step in trying to recover function in these developmental disorders. I think it's an exciting prospect.
Okay, so you put Shank back in the adults, and how long after you do that do you see increased social interaction? Because what question that comes to my mind, if it happens quickly, that suggests there aren't structural or major structural problems in terms of whatever dendritic arbors or synapse numbers, and rather it's more of a functional deficit rather than or a structural problem.
No, that's a great point. And we honestly didn't know what we were going to find. So SHANK3 specifically, it's responsible for creating this postsynaptic scaffolding protein. So exactly like you said. And again, it's not really well understood how it disrupts the broader circuit. Social behavior is very complex. It likely recruits many different parts of the brain to bring about social behavior. But this part of the brain, so the dorsomedial prefrontal cortex, is thought to be a central hub where information is likely routed through other parts of the brain, or there's some form of integrational process where you take different sensory stimuli and integrate them to produce an appropriate social type behavior. And so it takes a few weeks for once you reactivate the gene for the proteins to get re-expressed in the brain. The really interesting thing that we found was that as the protein was re-expressed, we started seeing slowly over weeks neurons starting to behave more normally, meaning that they were starting to encode information in a way that mimicked how normal wild-type animals encoded social information. And very shortly after that restoration of the neuronal function, we started seeing restoration of normal social behavior by almost a week. So first you started getting expression of the protein, then a slow restoration of neural activity, and then shortly after that, a slow restoration of behavior. And this happened over weeks. So it's not immediate. But honestly, it was a little surprising. We're very excited when we saw that happening. And what it suggests is that even in individuals that have had a lifelong deficit, and these are adult versions or the adult animal version, it is still potentially possible to restore functionality. So it kind of opens up the window to a lot of disorders that are developmental and genetic, and it suggests that there still is a lot that you can do, both from a neurological and neurosurgical perspective.
Yeah, and there's a lot of advances, and I've had multiple podcasts on non-invasive ways to increase or decrease the activity of neurons in, brain rate, whatever, transcranial electromagnetic stimulation, or maybe even direct current or something like that. So maybe, and you think about someone with autism, maybe is there work going on with that in people with autism doing that kind of non-invasive?
Yeah, actually, there are some studies here in the Boston area where people have been using TDCS or TMS, transcranial magnetic stimulation, to try to modulate these circuits. And there have been some early promising results. It's still very early on, and it's still challenging. One of the bigger challenges is that it's such a heterogeneous disorder. Everybody's a little different, and there's no one cause for it. And this is to contrast it with certain disorders like, for example, Parkinson's disease or essential tremor, where it's a fairly well-defined circuit with a fairly well-defined mechanism. There's still some variability between individuals, but we have a really good understanding of it. And oftentimes these symptoms can be treated fairly effectively with one or two types of treatment. But this is not as much the case in disorders like autism spectrum disorder.
Okay, and then you did some work on social decision making in humans too?
Yeah. So the interesting thing is, so there are a lot of things that you can do and questions you can answer in animal models, but there are a lot of things that humans do that are fairly unique to us. And so there are things like theory of mind, in which it basically means the ability to understand that somebody may have a belief that is potentially false, and not only false, but potentially different from my own understanding of reality. And so it's essentially our ability to predict what somebody else is believing, not simply what they're going to do, but what are their internal thoughts and beliefs, and understanding that somebody else's beliefs may be different from my own. We do this constantly, and individuals with autism spectrum disorder are often thought to have difficulty with this. But it's really fundamental to everything that we do. We're constantly trying to figure out what somebody else is truly thinking, even though we may, there's no way to really know what somebody else is thinking. And it's really important to human ontogeny, to our social development. And people who are very good at it, can do very well, both within their jobs and socially. Some are better than others. And it's something hard to ask an animal. You can't really ask them, what do you really think or feel?
Right.
There's some early hints that certain primates may have the ability for theory of mind, but at a very rudimentary level. So this is something that we want.
Ziv, I had one of my first podcasts. I had Nikki Clayton. Do you know where her work on the crows or the corvids, the members of the crow family? And she has this evidence certain of these species, they hide food, right? And she has pretty good evidence that, one animal, for example, they won't hide food when another animal is around. And if another animal is around when they hide it, then when the animal leaves, they'll move it.
Yeah, yeah.
And it's a lot more rudimentary than humans for sure. And of course, we evolved over time, so it's maybe not surprising that uh, this mind reading ability is something that's been evolutionarily very important for survival. Yeah.
No, that's absolutely true especially for social animals. And crows are unique. They have abilities that sometimes match many abilities that the humans show, including this cheating type behavior where they know when to hide, or sometimes they'll actually pretend not to hide until somebody is not looking, and then they'll hide it. So they have these really complex, interesting behaviors. So to a certain degree, we're all just evolved animals as humans. We show some of these interesting capacities. But yeah, those are really classic experiment. I really love that. But so, the interesting thing in humans is also you can use language to ask these really
Right.
complicated questions.
Yeah.
And so in this particular study, what we did is we used this classic task. It's called the false belief task. And it basically, it's a narrative that tells you a story about something that may be happening. So for example, you can tell a story like you and Bob are sitting at a table drinking coffee. Bob leaves to the restroom and you put the coffee from the table and hide it under the chair. When Bob comes back, where will he expect to find the coffee mug? And even though the coffee mug is under the chair, you're able to infer that Bob likely still thinks that it's on the table. So it's really complicated, right? But you're able to somehow take all these words, glue them together into this really complicated narrative in your mind, build a representation of where, of Bob, the table, the mug. This is all happening very rapidly. But not beyond all of that, you're able to still make this really complex inference about what Bob is likely thinking when he asks, what word does Bob think the coffee mug is?
Well,
And
the first thing I'd say is, Bob, what did you do with my coffee mug?
exactly. Well, that's an even higher level of theory of mind.
Oh,
So,
okay.
you've gone beyond the
Uh,
basic what we call first order theory of mind. But yeah, even beyond that, we can ask second order type questions like, what does Sally think about Bob's thoughts about the coffee?
uh,
And with these narratives, you can make all these really detailed manipulations. You can ask these kinds of questions across scenarios in which you're asking questions about a location of something or different events that have to do with bicycles or cars or different social agents. And so we can give these really complex narratives, and across all those narratives, ask a really simple question about what are neurons, individual cells in your brain doing? And are there certain cells in your brain that are finely tuned to tracking another's beliefs? And the intuitive answer to that would be like, of course not. That would be crazy, that you have cells that are that finely tuned
right.
to really focusing on somebody else. But we actually found a pretty prominent proportion of neurons, at least in these parts of the brain, the prefrontal cortex, that are fairly reliably able to predict what somebody else is thinking, even though at no point was it explicitly stated. We never said Bob's belief is false or Shelley's belief is true. But some of these neurons were fairly accurate. They were able to tell whether somebody else's beliefs were true or false. And beyond that, they were able to distinguish somebody else's beliefs from my own beliefs, or even another's beliefs from simply the physical state of reality, simply knowing where the cup is or the mug is.
Hmm.
So they were very uniquely tuned to trying to figure out what somebody else is thinking and believing, which was really exciting. I mean, that was honestly.
And of course, you're finding that these neurons are kind of a prediction of what others believing, but that doesn't mean there aren't other neurons that you're not recording from that are also involved in that prediction. So there's,
Yep.
you know, intuitively, maybe realistically, it makes sense that, you know, they're part of a bigger networks that's involved in that, right?
That is absolutely correct. Yeah. So we were able to only listen to this small part of the broader network of cells, and what's likely happening is all this information. These are really complex representations, and there are likely many other parts of the brain that are doing many different things, including holding representations of where the cup is, who Bob is, what your relationship with Bob is, who's doing what, and how to whom. But what we're doing is we're just listening to small parts of this broader network as information is rapidly flowing through them. And we can basically listen to them and ask, are there certain parts of this broader network that are uniquely tuned to these bits and pieces of information? But you're absolutely right. It's the whole brain or big parts of the brain are likely involved in this process, and we're just listening to a small part of it.
I think most people appreciate the importance of this work and how you're getting to answers that are not possible otherwise. But we often in multiple podcasts we've had my guest is does a lot of functional MRI, right? And so you can look at neural network activity at it's a very crude level from the standpoint of certainly individual neurons, but even neural circuits. So as functional brain imaging, the resolution is such that a pixel is only, I don't know, it's at least maybe 1,000 neurons and 10,000 neurons, 100,000. And it doesn't really tell you much beyond there's activity going on here. It doesn't really necessarily tell you. So having said that, is there any value of trying to correlate or somehow do the same paradigms you're talking about and get functional MRI data and then somehow see if you can tease out any relationship with what you're seeing in individual neurons?
Yeah, no, that's it. That's absolutely, the correct way to think about it. It's one of the things that we've been very interested in trying to do. So there have been quite a lot known about theory of mind and how you form representations of others through functional imaging studies. And one of the reasons that we had focused on parts of the prefrontal cortex thought to be at least broadly involved in this form of cognition. But they give you good insight into what are the areas of interest within the brain that may encode certain or may be involved in certain behaviors. But they also give you a much better network type of representation of what is happening throughout the brain as you're doing these really complex tasks. What we're able to do is to zoom in super finely at the resolution of individual neurons, and then ask questions about, well, at the very basic computation level of individual cells, how is this information encoded? And one of the challenges, and a lot of my good friends are, they do focus on functional imaging or other forms of broader kind of cortical surface recordings that don't have that resolution. And it's really exciting to get some convergence. We're seeing this really interesting convergence between functional imaging studies and single neural recordings. But you get information different resolutions. I think ultimately to really understand the brain, we have to be able to go all the way from the broader mesoscale all the way
Yeah.
down to the individual, individual computation level of individual neurons, which are kind of like the transistors of a computer. They're where all these individual ones and zeros are happening. So I don't think any one technique gives you the answer. It's really a combination of everything.
Yeah, that's pretty cool. Okay, language. This is no one really knows how the heck language works when you get down to individual neurons, groups of neurons. Most neuroscientists most neuroscientists, I'm one, know very little about how language works, except we've heard of Brokaw's and Wernicke's
Yeah.
areas, and we know that lesions of those, damage to those, can affect, uh, either speech or language comprehension. And you've been looking at both um, speech production and language comprehension,
Yep.
recording from people.
Yeah, no, language is such a fascinating thing, honestly. It's something that we do seamlessly. We do it all the time. It is in its uh, innate being really also a fundamental social construct. I mean, if it wasn't for other individuals or social interactions, it wouldn't really be a good reason to talk or to listen. So it is really a social construct, but it's also really complex when you think about it. So when I'm, for example, talking to you right now, you're able to somehow take these random sound sequences, these individual phonemes, glue them together into individual words, then take those words and extract some kind of meaning from them. So when I said g or dog, you're somehow able to take those random sounds, put them together, and like build a representation of a dog in your head. But even beyond all of that, you do this really rapidly. So most of us say about anywhere from 2 to 4 words a second. And at that really rapid temporal scale, you're able to not only extract the meanings of individual words, but you're able to also glue them together into phrases and sentence level representations, and build this really complex understanding of things based on these phonetic features and words. Um,
And in
so we,
conversation, so like right now, I was just thinking about, so there's kind of two conversations going on in my brain. One is
uh,
my conversation with you, and one is with myself,
right,
as you're talking,
right.
thinking about what you're saying and thinking, well, should I ask a question? What might be a good question to ask? And it's happening so rapidly. It is incredible.
It really is. And it's something you can really easily study in animals. There are animal versions of conversation. So there are animals like canaries or zebra finches that
Huh.
really have these extremely complex vocalizations, and even there's some syllabic structure to them, and they're very personal. But one of the first things that we really wanted to try to understand is how do you extract meaning from words? And there are tens of thousands of words, for example, in the English language. And they all have unique meanings. They are all embedded within sentences. And so it's a complex thing to ask. But one of the things that we try to first look at is just, are there certain neurons that are tuned to particular meanings? So for example, are there certain neurons that, like uh, uh, conceptual meanings such as animals or emotional states or people or food items, things like that. And so we can use these vectorial representations of words where we basically each word has, without getting into the details of how this is done, each word can be assigned a particular position in this broader semantic space across all words. Where, for example, words that are similar in meaning, like dog and cat, are closer together in this space compared to dog and chair, for example. And then you can group all these words based on their shared meanings. So, for example, you can combine things like carrots, salad, cherries, pears into, food type category. You can then group words like clouds, rain, wind into like a nature type category. And then you can ask, are there certain neurons that are tuned to these particular meanings? And the interesting thing that we found is that even within natural speech, just free-flowing natural speech, certain neurons would increase or change their activity very uniquely only when they heard certain words that had a particular shared meaning representation. So certain neurons really only liked emotional states like happy, sad, glad. Some neurons, and they didn't really respond to anything else. And then certain neurons really responded to actions. So like run, shake, grab, throw, which was really interesting. They were basically partitioning this information across different types of cells at a very fine cellular scale. So that was one of the first findings, and that was really exciting.
And this is still in the prefrontal cortex?
Right. So this isn't a part of the prefrontal cortex that is thought to be, at least to overlap somewhat with part of the broader language selective network. So these are parts of the brain that activate distinctly to language-related tasks or processes. But it's also thought to be very broadly involved in semantic representation. So whenever people have done used semantic tasks, in functional imaging studies, this area is fairly consistently lit up. So it's thought to be one of the areas that's very involved in semantic processing. So it was kind of interesting to see this really fine scale resolution or map of semantic space. How do you map out all these meanings onto the activities of individual neurons?
And you also had a study on, okay, so you had one, I guess these are both in Nature and this year,
Yeah.
right? One on speech production and one on language comprehension. So you were talking mainly there about the language comprehension.
Correct, yeah.
And then the paper on speech production, what question were you asking there, and how did you go about investigating that?
Yeah, so speech production is also a really complicated thing when you think about it. So let's say you think, let's say you know the word that you want to say. So I'm going to use a hard word like anti-disestablishmentarianism.
Yeah.
So you have that, word in your head, and somehow you're able to take all the individual phonemes. So a phoneme is something like a pa or a ka or a da. And you're able not only to have a full representation of the word in your head, you're able to glue them all together and do it in the exact right order and the exact right syllabic structure. So starting with something simple as the word dog. At some point when you want to say that word, what we found is that there are individual neurons in the brain in which some neurons will be uniquely active whenever you want to say the sound da, and another neuron will be uniquely active when you want to say the sound ga.
Huh.
But beyond that, there are these neurons that will only activate when you say da followed by ga versus ga followed by da. So dog or god, for example. And when you think about it, that's necessary to a certain degree. It's not enough just to be able to throw a jumble of speech sounds into a word. You have to say it in a very specific order and structure. So one of the first things that we found which was very exciting, is that there are neurons that are very tuned to these sound features that ultimately go through your mouth to produce a particular articulatory sound. But beyond that, there's a structure and mechanism within your brain that allows you to really rapidly shuffle all these sound sequences, glue them together into an individual word, and pump it out extremely rapidly over these sub-second time durations. And this is during natural speech, as people are like producing, 2 to 3 to 4 words per second. So this was happening extremely rapidly and in a very, very fine spatial and temporal scale. And so by being able to listen to the individual ones and zeros of these single cells, we were able to find this structure and this process that let people produce, meaningful speech, basically.
Yeah, that's incredible. And we have a we have a 1.5-year-old granddaughter. And unfortunately, my son and his wife live very close. So we actually see her. She comes over here 3 days a week. And so this is an example where she's in an age where she's clearly has a lot of words encoded in her brain.
Right. Yeah.
I mean, it's very clear. You can say a word and she'll point to it or whatever. Maybe hundreds of words by now. But as far as articulating speech, she's only got a few. One of the words she's learned a lot now is no, but she has only a few other words, right? So there seems to be, at that point, it would be really intriguing to know what's going on in those circuits at that stage.
Now, there's a lot of really interesting research on this. Well, one of them also has to do just simply with culture. So if, for example, she was born somewhere in China, then she would have a somewhat different representation of all these phonemes or these phonetic features, including the tonality. So we're not a very tonal language. English is not very tonal, and Mandarin is. And a lot of these tones have very specific meanings. And you will see individuals who grew up in a different country and they try to learn a different language. They will still use the same core phonetic features and way of articulating words that is really hard to unlearn. So there's something that's hard baked into the brain at some point as you develop. And then there's some really interesting studies, not using single neural recordings, but looking more broadly at coarse brain-wide representations. And there's a very interesting sequence of events that happens, including just the development of white matter tracks that connect lower sensory areas to higher articulatory centers and integrational centers. So there's a lot happening, and there's so much to learn about it. And it's a really exciting time to be in this field because there's a growing knowledge of how we develop language, how we structure it. But there's still much that isn't really known. So
yeah, that's a good point. It focused a lot on the prefrontal cortex, and from a developmental standpoint, it's generally accepted that it's kind of one of the last regions to fully mature. And you mentioned the myelination, the white matter, which enables fast conduction of impulses, and that. So you've got the prefrontal cortex critical for decision making, language. So maybe that kind of, but on the other hand, the language clearly develops way before the decision making are right. The idea is that even adolescents are poor in decision making, and
some adults do, but yeah. Yeah, there's a lot, and it's interesting because sometimes people treat kids or even teenagers as fully grown adults, but there's still a lot of development that happens even later on, in life, including, like you said, decision making. That's
yeah. And so what's your next big study? And I'm sure you've got a lot in the works. And so who's the first authors on most of these papers? Are they postdocs? Are they graduate students? A mixture? Or
it's a mixture, yeah. We have a phenomenal group of individuals. Many of them are from around here. Some of them are postdoctoral students, or just, yeah, absolutely phenomenal. That's honestly one of the most fun part of these things. It's fun being with other people that are very curious and open-minded and like to ask
Yeah.
very curious questions, or curious about how the brain works. But yeah, no, these are all large team efforts, for sure.
Yeah. That's great. Uh, let's see. And so as far as what do you see down the road as far as implications of your work? So you mentioned autism, right? Which there's very clear implications. What about for um, social groups, society? Uh, in theory, understanding the brain may enable improvement in human interactions, maybe
Yeah,
fewer wars, less
I
conflict.
think we need that.
I
Yeah.
don't know. We're
Yeah.
going to eventually have, be able to tune our brains so that they're more, um, you know, amenable to good social interactions.
It is interesting. I do think about that. I'd say probably two parts to your question. The first is, when work is this going, and what can we do with this knowledge? So part of it is using some of this knowledge to develop, novel treatments. So for example, I'm involved in a group that's very interested in treating patients that have paralysis, and for example, cannot speak from conditions like ALS, in which people are essentially locked in. Their mind is fully functional, but they're just not able to move or speak or communicate. And where we're creating synthetic speech, or allowing them to speak through an artificial brain-machine interface, where we read their thoughts and we're able to produce them in spoken format.
So this goes considerably beyond what Stephen Hawking, what
Right.
he used.
Yeah, he used I believe eye movements, and there are certain devices like Tobii device that can read your eyes, and you can very fairly effectively communicate in that way. It's still very slow and fairly coarse, but there are a number of groups including ours, that are very interested in trying to read information from the brain and use that to produce speech or movement. Within the social broader social spectrum of interests, there are groups, including ours, that are interested in using neuromodulatory approaches like deep brain stimulation to treat disorders like depression and social behavioral disorders, including autism spectrum disorder. But it's going back to one of your first questions or comments. I think one of the things about humans is that it's honestly some of these imperfections, what we would consider imperfections, that make us human. And I don't necessarily feel that you have to correct all of it. There are a lot of, and this is part of evolution. I mean, things even like aggression or strong emotions, these are part of, it's a fundamental part of who we are. And they all, to a certain degree, had some evolutionary benefit and reason. And it makes people are very different from each other. We're a very heterogeneous type type of animal, really. And I don't feel that we have to homogenize people so we're all super polite and all the same.
Yeah, that's a good variety is the spice of life.
Sometimes, yeah.
Yeah.
I'm super nice, I think, and very chill and very, but sometimes I've been told that's not a good quality. So, but
Oh
everybody's,
yeah, well,
yeah,
yeah, it's when you're in a leadership position, right? Then
sure, yeah.
yeah, 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 that's a good point. Well, it'll definitely be interesting to see how humans evolve, our brains evolve over the next whatever thousands of years.
Yeah.
My understanding is and this is based on measurements of skull volumes, that since before the agricultural revolution, at least by skull volume measurement, the size of the human brain has actually gotten smaller overall. And we're very specialized now. We're all specialized, right? We learn how to do something very good.
Yeah.
Right. And whereas a long time ago, the humans and hunter-gatherers essentially were very multidimensional in terms of what, and that one thing I'm concerned about is we don't move through our environment much when we're thinking, because we evolved moving through, like, we've got 2.5 acres, there's woods back here and a stream, moving through the environment and thinking about where we are, what we saw, and who we saw or whatever. And now a lot of us are just sitting in a computer. Maybe scientists more than others, I don't know. Certainly some occupations are kind of very repetitive.
Yeah.
I think we're kind of blessed in that respect, you and I, because we have the time and ability to expand our knowledge every day and do a lot of deep thinking. Yeah. But there's a lot of concerns I have with the way the internet's going, and kids, like my daughter in particular, she spent a lot of time on TikTok, and kids aren't reading books, I don't think, as much as they used to, and having a lot of quiet time to think deeply about things. But it'll be interesting to see what happens as we evolve with this technology.
Yeah, it's always a little challenging to find a causal relationship. We've actually been very interested in things like that, including even how COVID affected individuals and their social well-beings. It is interesting. I've thought about similar thoughts, and like you said, Neanderthals actually had, and earlier hominids had bigger, presumably bigger brains, partly. And again, it's not totally understood whether it's the fact that they were very carnivorous species, so ate a lot of meat, so you have a lot more calories to work with.
I don't know about that.
Yeah. Maybe not these.
Yeah.
But they also needed to adapt. They had to be able to adapt to everything, and but we also have culture now, so we are able to rely on past generations and the accumulation of knowledge to do things that was absolutely unthinkable, even 1,000 years ago. But even brain size, it's a hard metric to say, because like you said, even crows, they have relatively small brains, but they're incredibly intelligent. They can do things that most canines would not be able to do.
And their brains organize very differently than ours too.
Right, yeah. So intelligence may be a little different. It doesn't necessarily just purely scale with size. But there's a lot of interesting questions. I mean, it's hard to tell where we as a human species are gonna, what trajectory, where it's gonna take us. But it's fun to think about it.
And whether, I know your time's precious, but another question is, will we remain as one species?
Right. Yeah. Yeah. If you read
evolutionary history, usually that doesn't happen.
It's interesting. Yeah. I mean, if you're a Herbert fan and you've read Dune, that's an interesting kind of like how humans may speciate in over tens of thousands of years. But it's it's interesting. Yeah.
Okay, Ziv, thanks for your time. I appreciate it. I enjoyed our conversation. It's fantastic work, and
thank you.
Really, really important and valuable, not just for science, but for humans down the road in particular.
That was a pleasure. Thank you so much.
Okay. Bye.
Bye.